Hello all. In this session we are gonna see about after clipping the grids, what are we gonna do, where are we gonna transfer the grids and how it's gonna go into the microscope. Initially, there are certain components we have to be aware about before using the loading station. Let's have a closer look onto it. Initially, whatever you are seeing is the loading station. Now we'll look into the individual components and what are they. First thing is the grid box transfer pedestal, and here is where there is a groove into which the grid box will be getting into. After that, the middle portion is where the cassette will be loading into it. So this is how the dry look poster of how this grid box transfer pedestal looks, and it works. The second component is the cassette holder or the cassette ejection wand. Basically, if you see here, there is a arm which is gonna hold the cassette. This arm which is helping to hold the cassette when it's transferring to the further component which is going into the microscope. The next component is the exit hatch cover, which will be going on to the loading station, and the other foam wares and the plastic cover which close the entire loading stations. Now let's get into the assembly part of it. Now looking onto the closer look of the loading station, the main groove which you are seeing here is where the grid box transfer pedestal will be sitting into, and through this position we'll transfer into the nano cap, that is the other component which will be going into the microscope. Now let's assemble the individual components one by one. Initially starting with the grid box transfer pedestal, which will go into this specific portion. Followed by the cassette. Now when you look into the closer view of the cassette has individual 12 grooves where the grids can be placed into. Basically, the groove which you are seeing here is where the cassette holder will grip into, so that the cassette can be moved from the loading station to the nano cap. Usually make sure the silver portion should be towards the other side and the golden portion towards to be you. So we are keeping the cassette here. Once the cassette is being kept, next we will keep the cassette holder. This portion will go into the base of this. Basically, this is the portion which is gonna help to transfer the cassette from here to the nano cap. The next component is the exit hatch cover, which basically goes into the side of the loading station and gets locked into it. The entire mechanism of the bottom portion we can see in the further part of the video. The next two components are the foams, which will basically settle on this edge and this edge of the loading station, followed by the central plastic cover which covers this entire station. Thus, the entire component assembly of the loading station is being done. Now, let's look on to the cassette and its internal overview. If you look onto the cassette, as we said before, it has individual 12 grooves onto which the grids will be getting into it. Now let's look on to the line of it. Basically, the cassette's groove is something like a L-shaped bridge into which your grid is gonna be sitting into it. So the grid won't be supported either on the bottom or on the top, but on this L shape where the grid will be getting supported into. So it has been very crucial that the grid has to be properly placed into this L groove. Without that, your grid will fall on to either the bottom of the cassette, which will ruin your entire grid as well as the cassette while taking it out. So it is bit more essential that you should have more concern on looking where you are placing the grid into it. Now we are going to see how to transfer the entire grid into the dry condition. So this is the clipped grid which is in the grid box, holded by the grid box top fastener wand. Now we'll take this thing into the loading station. On the top view, you can see how the grid box is placed into the groove. Now we are basically loosening the cap of the grid box. Once the cap is out, you can see the clipped grids which is placed into the grid box. Now let's take the flat T autoloader tweezer with which we can take the grid from the grid box and place it onto the groove of the cassette. This is how we are gonna place the grid, once clipped grid. This is how we are gonna place the clipped grid into the cassette. I'll also demonstrate another one. We are gonna take the grid, clipped grid from the grid box, taking it out and placing it between that L groove. Now you can see the L groove more clearly here. Between this L groove, you are gonna place the cassette and gonna tighten it using the Fast-T autoloader tweezer. Once you have placed the grid into the cassette, there is also a checking step which we follow here. That basically we are gonna tap the grid using this tweezer to check whether it has been properly inserted on the top, also on the side very slightly to check whether the grid has been properly inserted into this groove. If at all it's not been properly inserted, this tap will loosen the grid from the groove and the grid will fall into it. So this will prevent the grid getting fall down when it is into the microscope. We are doing this here to have a clear visual onto the dry condition. Now we have seen how to load the grid into the cassette. After that, this cassette will be transferred onto another component which will be going into the microscope. That component is called as nanocap. It is also called as autoloader transfer canister. For a simpler term, we'll go with nanocap itself. If you look at the closer view of the nanocap, there is an internal groove onto which the cassette will be into it, and there is another pin which will be recognized by the microscope when it's getting docked into the microscope. Before every insertion, it has been mandatory to check whether this pin has been further functional. This is the exit hatch cover which we were explaining before. Now we will see how the nanocap will be placed into that. Before that, we have to loosen this switch and we can take out the exit hatch cover. You have to notice this flat edge here and the curved edge here. This is almost the same shape as of the nanocap. If you see the nanocap here, it also has the same thing of flat edge here and a curved edge here. We are going to keep the nanocap in such a way it placed into this groove and we are going to lock the session. This is how a nanocap will be clipped into the loading session. Now we have seen how to dock the nanocap into the loading station. Now we'll see how we are going to hold the cassette using this cassette gripper. Apart from that, how the cassette will be going into the nanocap. Initially, we have to move this cassette arm accordingly, and we are going to hold the cassette using the clip which is on the side, which this clip is going to hold the cassette. With this condition, we are going to take it up and we are going to put it into this nanocap. This is how the transfer of the cassette to the nanocap has been done. This process, whatever we are going to do here, has to be done in a very faster manner so that the grids whichever is in the liquid nitrogen can remain the same. If this process is being prolonged or if it is heated somewhere, this can cause the prolongation of the grid outside the liquid nitrogen, which will increase the chance of getting a crystalline ice contamination. Now on the side view, we'll see how the cassette is getting into the nanocap. Basically, we are going to take this cassette using this holder and we are going to drop the cassette into the nanocap which is here. So basically, we are going to take this cassette from here and we are going to drop it into this nanocap. Once done, we are going to take this out. Now if you have a closer view here, you can see the cassette which is into the nanocap. Now we have seen the entire process of how the loading station is going to work and how we are going to load the grid into the nanocap. We have seen all this entire process in the dry setup. Now we'll add the liquid nitrogen and we'll see how we are going to do the entire process under the liquid nitrogen condition. So initially we are taking out the safety cap of the plastic lid. and we are gonna pour the liquid nitrogen into the loading station. We have to pour the liquid nitrogen in such a way that the cassette is being submerged under. Now we have transferred the clipped grid into this loading station, and the station has been now filled under the liquid nitrogen condition. For usually we have to work under this condition while taking the grid out and loading it into the cassette. Since for your visualization we are just removing this entire cap out so that you can see the things in a much better view. So now you can see the clipped grid and the grid box here. Now initially we are gonna equilibrate the pen so that it comes back to liquid nitrogen temperature. Once the bubbling is being done, then we can further proceed with opening the grid box and we load the grid. Now we can see the bubbling has been stabilized. So now we can use this to open the cap of the grid box. Once the grid box cap is open, now we can visualize the grids, clipped grids inside. Now we can use the flat T autoloader forceps to take the grid from the grid box into the cassette. Before that we'll equilibrate it also to the liquid nitrogen condition. Yep. Now the bubbling has been stabilized. You can also use a screwdriver to hold the grid box while taking the grid out, because sometimes when we take the grid out from the grid box also either comes along with it. To avoid that, you can either use this screwdriver to hold it in such a way that the grid box doesn't pop out. So you can take the grid outside from the grid box and drop it into the cassette. For your view, we are doing it again. We are gonna take the grid out of the grid box. The screwdriver is gonna give us a support trim so that the grid box doesn't come out. Once the grid has been taken from here, we are gonna place it in the cassette where the L groove is, and we are gonna check whether the grid has been placed properly by tapping on the top and followed by a slight tap on the sides to make sure that it's been properly gripped into that cassette. It has been advised to load this cassette can hold around 12 grids as we said before, but it is advised to keep only the 11 grids, that is keeping the first slot as empty and loading the grids onto the rest of the 11 slots. This is because if at all there is a grid inside the microscope, that can come back to the first position of the cassette. Now we have loaded the grid under the liquid nitrogen condition into the cassette. The next part, while doing this process parallelly, you have to cool the nano cap so that once the grid has been loaded onto the cassette, it can be loaded onto the nano cap, which can be further going into the microscope. So for that, first we'll cool this nano cap. The same principle, until the boiling is being stabilized, it has to be cooled. See, now it's been getting cold, so the boiling has been stabilized on the surface. So this is how it has to be cooled once you have poured the liquid nitrogen. Now this cassette has been ready to be docked onto the loading cassette. Further we will transfer the grid. Now the nano cap has been cooled down, so we will now dock it to the loading station. In the dry condition, you have seen how we have docked it. The similar way, we are gonna remove the exit hatch cover and we are gonna dock the nano cap here and we are gonna lock it. This is how we are gonna dock the nano cap into the loading station. Once we have docked the nano cap into the loading station, now we can transfer the cassette to the nano cap. For that, we have to remove the top cover and all the foams which is supporting it so that the transfer process will be easier. So now you can see the entire station here. So this is the pin, this is the clip which is going to hold the cassette. You're going to move this arm here, lock the cassette, and the bottom is where the nano cap is. We are going to quickly transfer it into the nano cap. Once done, you can release this arm out. Now we can see the cassette docked into the nano cap. This nano cap will be further going into the microscope. Before transferring that, make sure once the cassette has been inside, the pin whichever you have seen here will be slightly enlarged, denoting that the cassette is inside. And also you have to, before docking it into the microscope, you have to make sure that the pin is still functional by just pulling it out and checking it. And make sure to add liquid nitrogen onto the fill. And now make sure before docking it into the microscope, you are filling it back with the liquid nitrogen. Once we have loaded the cassette into this nano cap, we have to take this nano cap with the liquid nitrogen and we have to just push it into the microscope and click dock. And during this situation, you have to be more careful about which orientation you are keeping the nano cap. This flat edge, whatever you are seeing, should be on the front end, it's facing towards you, and there is a pin inside this nano cap that has to be also shown. Now we'll just dock the cassette, now we'll just dock the nano cap into this microscope. Once I kept it into the microscope.
Explain.
Now once you kept this nano cap into this microscope, you will get a dock option enabled onto the screen of the microscope. Once the dock has been enabled, you can see the entire nano cap, the front part of the nano cap getting into the microscope.
Here you can visualize the autoloader grabbing the cassette from the nano cap to the microscope. Once the entire cassette is taken into the microscope from the nano cap, there is an arm which holds the cassette so that it can begin the next process.
Once the docking is complete, you can just remove the nano cap and do the rest of the process.
Hi all, in this session we will see about the interface of the software and the data collection. So initially when looking onto the software, the first part where the setup comes, where you will get an option for opening and closing of column valves, where in this vacuum overview you can see when the column valve is getting opened, here the valves are open so that the electron beam passes through the column, and when we close it, similarly the valves getting closed. And here you have a high tension. This is of a 200 kV machine, so you can see the emission current and the high tension values here, and here the FEG values, you can see what is the exact voltage of FEG is flowing through the circuit. And apart from that, in the autoloader section you will see the total number of grids. Here you can insert around 12 grids all at once, and you can whichever is in the blue tick mark are the grids which is already checked, and the yellow one is the grid which is in the microscope. Here you can load one grid into the microscope at a time. And apart from that, here you have a temperature control. Since it's a cryo temp, you can see the liquid nitrogen percentage for the individual stuff. And here on the search column, you can see you can have a overview of the grid where you can the blue cursor shows at which position the stage is right now. You can either add or delete the values so that accordingly you can store the places where you can come and screen it again. And apart from that, here you can see the spot size varying from 1 to 11. Usually for the cryo-EM, we used to keep the spot size at sometime 8. And for the imaging, we use a nanoprobe mode. And apart from here, you will have an options for the stage whether to reset the holder and also to turn on the wobbler, etc. And on looking onto the bottom, where you can see the magnification, here we are at 45,000x, and apart from that, the mode which we are imaging, that is a nanoprobe, and the high tension value here, the individual spot size, and here you can see the XYZ value where the coordinate of the stage where it is right now, and the dose rate what is currently going on. So now let's go on to the next software to see the interface and how we do the imaging part. So we have seen an overview of what are the things there in that the other software. This software, whichever you are seeing right now, is a latitude from the Gatan. Here we will see a rough idea, like we will see a rough overview of how the imaging is happening from using a low dose function. So initially we have created a very small atlas having all the different types of eyes for the screening purposes. So I have selected one square, and here you can see the individual holes. So let me add a couple of holes for the screening. So here we have a colored denotation to understand the status of the imaging. The yellow color denotes that we have added the hole. So once you give schedule, it turns to green, indicating that it's in the track, and once it's completed, it will move to the blue. So once you have scheduled the things, you can see that this part, the hole which you have selected, becomes active, and the stage moves to that specific location, and it does the focusing, and follow up it will do the imaging. So once it has turned blue, saying that it has been acquired, now we can see the individual hole, and where I have kept the focus on the carbon area, and I've added 2 images for the imaging. So once the image has been acquired, you can see the dose and what mag it has been collected and what is the defocus. And on the next to the screen, you can see the live FFT of the image which has been already acquired. This is how the image acquisition and the data acquisition happens in the case of Cryo-TEM.
So apart from the software we have shown before, there is another palette here which we use for changing the intensity, magnification, spot size, etc. If you see this entire panel, you have a trackball here to track the beam, like to shift the beam to the center of the stage. And apart from that, you have a intensity knob where you can change the intensity, and there is a stigmator column to enable the condenser and objective stigmator. Apart from that, you have a magnification knob where you can change the magnification, a focus knob, and there is a multifunction X and Y which you have seen it in the theory classes. And apart from that, you have a eccentric focus, and here to adjust the eccentric height we use a Z here, that is the options are given here. Apart from that, you have a joystick to move the stage during the screening process.
Hi everyone, today is the last day of this entire course, and I covered most of the theoretical part of this electron microscopy and cryo-electron microscopy and image formation, different types of sample preparation, and little bit about the image processing also. And my students and I also involved and taught you guys how to implement those theoretical part in a research laboratory, how to prepare sample, how to collect data, how to image. And when you are imaging, you can see different kind of artifacts which are connected with the microscope. During my class, I taught you all of these things, and here also you can see all of this, experienced everything here. And I hope you enjoyed the entire course, and see you all later.